I should say that Cetero went out of existence, I think more than a year ago, if any of you are aware of that name, so I must not have filled the forms out properly. Topical drug products have been a problem in terms of demonstrating bioequivalence since, for the most part, clinical trials have been required by the FDA since 25, 30 years ago. It's really hard to measure blood levels of topical drug products for most of the topical drugs, as well as the FDA having sort of a bias that they weren't sure whether drug levels were really that meaningful when you're talking about topical products that don't act through their systemic action. But their interest in this area, since they do want to get to surrogate tests, due to the relative insensitivity of clinical tests and the high variability, they have stimulated a lot of work that now goes back over 25 years. And a number of surrogate tests have become available, well worked out and partially to fairly completely validated, that we're at the point now where it's pretty easy to see that surrogate tests are going to be available in the near future, and the FDA is actively working on this. This is a biopsy of skin, and skin's right in front of us. This shouldn't be a big problem to determine what's bioequivalent and what's not. And really, it's relatively thick. Over most of the body, this could be anywhere from 1 to 2 millimeters. The barrier layer, stratum corneum, you see here, this kind of homogeneous area right here is really the epidermis, the living part of skin. And then most of this fibroelastic tissue, which doesn't have a lot of cells in it, but that's where the circulation is. We really have a lot to work with in terms of surrogate methods, and show that schematically here. FDA has been interested in developing an entirely in vitro release test, both for transdermals and topicals, equivalent to the dissolution test that's done with oral drugs, which just looks at release from the formulation. It really has nothing to do with the skin. So right now, we don't know what the status of this is. They've been hot on it for a number of years and not so hot presently, although it probably is used now as not as a release test, but kind of a quality control test, batch-to-batch quality control. But in terms of surrogate methods, we have the ability to look at the barrier layer by simply tape stripping it. It's considered to be like the bloodstream if you're thinking about an oral drug, because if drugs don't get into this layer, this dead layer, to create a concentration gradient for diffusion, then you're not going to get much drug going through here. So it's really looking at partitioning of the drug from the formulation into the skin. I'll talk more about measuring a pharmacodynamic test like transepidermal water loss, or looking at exfoliation. Some drugs actually make the skin scale, so that could be a pharmacodynamic test. Skin irritation, which is required of all topicals anyway, has never been thought of as a potential surrogate test, but for some drugs it could be used that way. Vasoconstriction, that's the only surrogate test currently available, approved by the FDA for a whole class of drugs, the non-inflammatory corticosteroids. So that's out there. Microdialysis, where we look at concentration in the dermis, very close to the bloodstream, has a lot of interest currently, not fully validated yet, but what validation there is looks very good. This test is really the cornerstone of what I think will be a group of surrogate tests that will be used in the future. This involves the use of cadaver skin or surgical skin obtained from tummy tucks or breast reductions, or from a burn center, people that donate their body and have skin taken and dermatomed, cryopreserved, saved in a skin bank, ready source of skin. It's an in vitro test, the in vitro permeation test. And the nice thing about it is that it looks at all the steps in the absorption process, the partitioning, the diffusion, and then in vitro and vivo correlation shows that the rate at which drugs get through the skin is really equivalent to the rate at which they get into the bloodstream. So it's a built-in toxicity test. If the same amount for a potential generic drug is shown to be equivalent to the amount absorbed with the reference drug, by this test, you know the systemic blood levels can't be any different, and immediately you rule out any concern about systemic toxicity. Now that doesn't tell you about local toxicity, and that's why skin irritation is one of the tests that's required there. But our view is that all of these tests, people can criticize and say they all got limitations, but they don't all have the same limitation, and therefore you can select a couple of tests that complement each other. And I'll just show you what we've done over the last few years that kind of suggests that this is a valid approach. Well, the in vitro permeation test is one of these relatively low-tech tests like most of what we use for the skin. Basically you get a piece of cadaver skin and maintain it at body temperature with some sort of saline receptor here. You leave this exposed to the atmosphere and you just apply your drug here at a relevant clinical dose. Or if you're talking about a transdermal, you just put the patch on top of it. So it's good for transdermals or topicals. And then you just measure the rate at which the drug comes through by simply sampling here and analyzing it by originally LC, but now of course you have LCMS and LCMS-MS. So the analytical tools have gotten much better. So it's a simple test, the key being apply a relevant clinical dose, no problem for a transdermal because you've got a patch to work with, and you just maintain the skin at body temperature. The sensitivity is why I think this will be the cornerstone of a group of surrogate tests that will be used for demonstrating bioequivalence. This can also be used in the drug development stage of new drugs because it's a good way to compare the bioavailability of potential prototype formulations. But this was a study that was actually done to develop a generic drug very many years ago, the antifungal ketoconazole, the RLD being Nizoral, and this being its rate of absorption through the skin in terms of micro square centimeters per hour over I think it was a 36-hour period here. And the generic company that was trying to match this RLD in reverse engineering couldn't quite decide what the levels of two co-solvents were, the propylene glycol and the isopropyl myristate. So they just made some guesses and made some test formulations which we compared to the RLD, varying in propylene glycol concentration from 19% down to whatever that is, these three levels here. And of course, none of the formulations matched, but they figured, well, it's got to be between this propylene glycol and this propylene glycol. And so they then made a series of other formulations, and in fact, one of them matched pretty good. Propylene glycol of 16% and 4.2% isopropyl myristate. But you can see we're talking about determining differences in ketoconazole absorption that are only dependent on a few percentage changes in propylene glycol and a little bit of IPM. Well, there's no other surrogate test I've seen that has this type of sensitivity. So this is the main reason I think that this is the cornerstone of surrogate methods. And what I'm going to show now is just three examples of how this could be used by a regulatory agency. Topical hydrocortisone used to be a prescription product, and I don't remember, 15 or 20 years ago, the FDA decided it could go OTC if it was 1% or lower. If it was above 1%, it had to stay prescription. So we took a series of OTC products, just bought at the local drugstore, and compared them all. And we had the original prescription product, high-tone cream, and the 1% high-tone cream is right here. And then we're going to compare it to an OTC product, cortisone with a Z at 1%. And as you can see, they differ by about, I guess, six-fold roughly in terms of their rate of absorption. So it presents a good test of surrogates to show that these are in fact inequivalent. The in vitro permeation test says they're inequivalent. So now we're going to pick a couple of other surrogates and see if we get the same answer. And notice that the big difference is up here at around 8 to 10 hours. So we're going to do tape stripping, which was a method that FDA thought would be a one-size-fits-all. It could be used for showing the bioequivalence of all topical products. And we're going to look at comparing the results of tape stripping with these results here, and we're going to take samples at 8 hours, and then we're going to take it down here at about 2 hours, and then out here at about 16 hours, and we're going to see what tape stripping says. Well, tape stripping is really low tech because you just get volunteers, you dose your products here, you put your drug on in a relevant amount, and then you go to the store and buy some Scotch tape, or we use a different, I can't even remember what tape we have here, Transport, there it is. That's one of the common, there's a couple of commonly used tapes. And you just tape strip them any time. The theory being, if you tape strip the stratum corneum, that's like sampling the blood. That is the barrier that the drugs have to get through to get to their active site, which in most cases, not antifungals, not sunscreens, but in most cases, have to be in the epidermis or dermis. That's where we're trying to hit. So you take these strips, and here we're taking, let's see, yeah, 22. Here we're taking 22 tape strips, which gets you, in most subjects, anywhere from 40 to 80% of the way through the stratum corneum. That's one of the sources of variability in this method. So we're going most of the way through the stratum corneum. And we're combining these strips into 2, 5, 5, 5, and 5. 2, you know, on the surface, you're going to get most of the unabsorbed drug. And then as you go in, you figured, well, here we should start it to get drug that really is in the stratum corneum and not just in the wrinkles and furrows of the skin. Well, you can see here when we compare the two products, which should differ at 2 hours, and we're going down 22 tape strips, there's no difference between them. Well, even at 2 hours, the in vitro permeation test saw a difference, but it wasn't great. But what happens out at 8 hours, and now you begin to see, not in the first 12 tape strips, but as you get beyond and into the last 10 tape strips, you begin to see the drug product that was more bioavailable, the high tone, begin to give you a higher stratum corneum drug level, which is what IVPT said. You know, if you don't get more into the stratum corneum, you can't get more through the skin. So we got more through the skin, we should have got more into the stratum corneum, and that's in fact what we did. But the naysayers say, well, you know, I still don't believe this. Maybe it's all going through some pathway that has nothing to do with efficacy. So we said, well, one assessment of efficacy, and I won't go through the data, but for 30, 40 years, dermatologists have known that there's a relationship between clinical efficacy and the vasoconstrictor assay. And that's the reason FDA has accepted the vasoconstrictor assay as the only method you have to do to show the bioequivalence of the corticosteroids. So we're going to see if more got into the skin on the outside, more got through on the bottom, then there should be more in the middle to produce greater vasoconstriction. Well, to do that, we had to do one other tape stripping test, and that's out here at 16 hours because for hydrocortisone, the typical vasoconstrictor test goes for 16 hours, and you also have to hydrate the skin because it has such poor vasoconstrictor potency. And so we showed that whether you do not hydrate the skin or whether you do hydrate the skin, at 16 hours, we can still see a difference between stratum corneum drug levels. So the vasoconstrictor assay done with hydration isn't going to change the results at all. And what you see is what you expect. High tone, which is the more bioavailable product, gives you greater vasoconstriction than cortisone. But these differences are just minuscule and statistically just barely make it. So, good validation, these three tests all say the same thing. Well, another interesting group of compounds are hydroquinone, which are widely used to lighten the skin, particularly where you have post-inflammatory pigmentation. And they're available, again, both over the counter and as prescription products. Below 2% or below, they're over the counter, and above 2%, they're prescription. Well, here's a sampling of prescription and non-prescription products, and as you can see, they're all over the place. So we decided to take two prescription products, one showing the greatest bioavailability, Melanex, and the other one, which I can't even see here, get that from the next slide. The other one, Hydroquinone Forte, which was one of the original products that probably goes back 30 or 40 years now. But look at their rates of absorption here. Peak at, I think it's about 4 hours, much, much greater than the Hydroquinone forte peak, which is way down here. Even though this concentration is only 3%. The interesting thing is, so again, IVPT says these two products are inequivalent. The interesting thing about hydroquinone is that you're treating cells, the melanocytes, right at the dermal epidermal junction. And it's very convenient that suction blisters separate at the dermal epidermal junction. So as a surrogate test, why not look at the extracellular levels of drug right at that level? So we produced suction blisters in a number of volunteers and got exactly what we expected. At 4 hours, here's the more bioavailable product, and here's the less bioavailable product. Again, showing that IVPT not only shows you what gets through the skin, but I've never seen an example where it didn't show you what got into the skin. Theoretically, the biggest objection is, well, how do you know things just by chance for your drug wasn't going through the follicular pathway and wasn't really getting into the skin? As if the follicular pathway was a solid pipe right from the follicle to a blood vessel. I mean, it's a ridiculous argument, but we hear it all the time. The follicular pathway is a long pathway. It's just another access route to the dermis. Some of it may go right to a blood vessel, but it's still just another access route to the dermis. So here's another case of IVPT and some other surrogate method, in this case a very relevant surrogate method, giving the same results. The last one is again kind of an interesting one because it also gets into the mechanism of action of retinoids. Retinoids were first approved for the treatment of acne, and now some of them are also approved for the treatment of aging skin, to kind of a wrinkle reducer, as well as psoriasis. So what we're going to do now is look at the correlation between the in vitro permeation test and a pharmacodynamic assay. It's really skin irritation, but with transepidermal water loss added to it. And the reason it's so interesting is this. This surrogate test is actually an indirect assay for the mechanism of action of tretinoin in the treatment of acne. What happens is this. When you apply over a 21-day period retinoids to the skin, and you look at transepidermal water loss, and you also look at peeling, which is this green thing here, green line, you are looking at what happens, and this is again done just on the forearm of volunteers, but you're looking at what happens to acne patients. Because when you first start treating acne patients with tretinoin topically, you don't see much happening in the first week. And then somewhere in the second week, they start to get red and scaly. And before dermatologists knew how to handle the drug, they thought that was part of the treatment effect. You had to make them peel like mad to get rid of all that dead skin and all those plugs that were causing the inflammatory reactions. Well, you see the same thing on the forearm. And what happens is this, that after about, in this case, we're using high-strength retin-A, so it takes only about 10 days in the average patient. You begin to see that scaling, which is disruption of the barrier layer. Well, if there's disruption of the barrier layer, which keeps our water in, all of a sudden, at the same time, you see this great increase in transepidermal water loss as measured with a simple instrument that's readily available. So, this is a great pharmacodynamic test because not only does it show great differentiation between various retinoid products, it's actually relevant to the mechanism of action of this particular drug. So again, we just put areas on the skin and mark off areas, and every day the subjects come in, we apply the drug. We have to put saran wrap on for a few hours to make the permeability of the forearm like the permeability of the face, because this is a high permeability area. They come back every day, we measure transepidermal water loss, we look at the skin and grade the scaling. And what you see, again, for transepidermal water loss is if you look at the placebo, you don't see much happening. If you look at the 0.01% retin-A gel, you see a little bit of increase in transepidermal water loss. And then if you look at the 0.25% retin-A gel, you see much more. So it is very, very dependent on what concentration you're using, which is great because you want to be able to differentiate one concentration from another. So it's kind of an internal validation of the sensitivity of the method. Well, I won't go through the other tests. I won't show the in vitro permeation data. I won't show the tape stripping data because we got the expected effects there. I'll just show you what the transepidermal water loss and what the assessment of peeling look like. And the reason I'm showing this is because whereas the first examples I showed were just interesting sort of validation tests with no particular regulatory implication, these studies were actually done with 36 subjects in each of the studies for the low strength and the high strength. And they were actually done and submitted to FDA. They were done under GCP conditions and were actually done with the hope that FDA would accept this data in lieu of a clinical test. Well, of course, that didn't work. But the data, the studies proved very interesting because the company had to go out and do the clinical test. And of course, this is one thing the FDA always asks for in a surrogate test. Well, give me some clinical data, that shows that what you're seeing in your surrogate test agrees with what you see in the clinic. Well, it not only agrees, we've got the internal validation that we can see the difference between two concentrations of retin-A. So what you see here is that for, if you look at TEWL, the generic, so the innovator, the RLD is here, and this is the innovator product. So no difference, in fact, between the 0.01% generic and innovator. No difference between the 0.25. If you do the classic confidence interval, they show as between 80 and 125. So clearly these would meet FDA standards to show that these two products are in fact equivalent to each other. So where I think we are now, and the FDA is very aware of all this data, and they're very interested, particularly since the generic companies are now paying money to get more manpower to the Office of Generic Drugs. The Office of Generic Drugs is moving ahead rapidly, and I think we'll see some draft guidances probably coming out in the near future. So our feeling is that the assessment of bioequivalence of topical drug products can be accomplished through the use of appropriately selected in vitro and in vivo surrogate tests. We know that all these surrogate tests don't have limitations, but the result from one test can very nicely complement another and confirm results. And I think the selection of these tests would depend upon the complexity of the topical formulation and whether the tests were Q1 or Q2 equivalent. And I think we've seen evidence of that in the last couple of years. Surprisingly, the FDA put forward a guidance on one product, a cyclovir ointment, where the only thing you had to do, in addition to some laboratory tests like viscosity and particle size measurements and that sort of thing, the only test you had to do rather than a clinical test was IVRT, the in vitro release test, and you can get generic acyclovir approved, and there's already 2 generics on the market since that guidance became available. Now, I don't think IVRT is going to be, here I kind of overplay it here because I'm sensing the FDA is getting a little not so positive as they were a few years ago on it. It is going to be something all generic companies are going to have to do, but it's mainly going to be for quality control and possibly in the near future for batch release. But we just don't know at this point. So with that, I'll end, be happy to take any questions.
So for the water loss, what's the meaning of the water loss correlated, right?
What it shows is that there's been a change in the differentiation of the epidermis. So the tretinoin, it's known to get into the nucleus and combine and then have its effects on RNA and new proteins and stuff. And basically, it's changing how the stratum corneum, which is our barrier layer, is built. So if you produce a change in the barrier layer, it's likely that that change is going to be reflected in a change in transepidermal water loss, because that's the prime purpose of the barrier layer. For us to live outside of water and not carry 5-gallon jug of water with it all the time. We have to have a tight barrier and restrict water loss. So it's an indirect measure of the retinoids getting to the epidermis, the living cells of the epidermis, binding to nuclear receptors, and then all the changes that occur subsequent to that, which is what its mechanism of action is in acne.
So this water loss only applicable for this particular?
Those. Yeah, now it may be applicable, there may be other drugs. For instance, salicylic acid or lactic acid, which are used for trying to produce scaling. I would suspect that it would also detect changes in those drugs, if formulation changes in those drugs. I don't think anybody's looked at that yet. Also, 5-fluorouracil, which is used to treat precancerous lesions on the skin, produces a lot of irritation, and because it's destroying these epidermal cells that are precancerous. So you do get a lot of redness and scaling. The question is whether the surrogate test would pick those up soon enough. So that hasn't been done, but at least I think those two other therapeutic areas, 5-FU or any drug that's used to treat precancerous lesions, I think could possibly be used this way.
I have another question.
Yeah.
So in one formulation, you see the dramatic difference in the vitro absorption for the different PD level, right? So one question is, if one you done with the PD solution, the other one is make a transdermal patch, and you see absorption for the PD, but you don't see absorption for the transdermal patch. So what do you think is the constant?
What is the cause? Couldn't answer that. I mean, is this, I can't think of the terms, you know, the original Alza patch are now these new things that don't have the solution. You're not solubilizing the drug first in something, but you're using, what's the name for that, where you just have polymers and, well, whatever. I mean, whatever the type of patch is, if you were using the same formulation and putting into an Alza patch, where you're literally putting the formulation in there, I would expect that you'd get the same result. If you're using one of these other transdermal systems, then I wouldn't particularly think you'd be getting the same results.
Did you have one more question?
So how do you apply the topical formulation to the skin in order to correct red dosing and the uniformity? Because I imagine this would be very important if one to read between different tests.
Yeah, this is one of the sources of variability. What we do, what we have, I mean, obviously if you have a liquid, it's less of a problem. So the big problem is the semi-solids, the creams and the ointments. And of course there's these positive displacement pipettes, so that's how we're applying it. But we take the top off the chamber, so we have full access to the skin. And when you take it off, there's an indentation that the O-ring has produced in the top part of the chamber. So you can see the area of skin that's actually available to your dose, and likewise is available to the receptor solution here. And some people, just with the glass tip of the positive displacement pipette, will just make sure they apply it all the way around. Now, it's never going to be perfect. And if you're off by, I can't remember the calculation, one of my colleagues did the calculation, by a teensy amount, it can easily give you a 15 to 20% error in your area. So you have to accept that that's going to be part of your problem. The fortunate thing is that we all differ by an order of magnitude. So that error looks pretty small when you're looking at different people. So, it's a problem, but you just do your best.
All right, so thank you again.
Okay.